Thank you, Pat. It's wonderful to be here. Truly, this is a spectacular campus. I have a thing about educational institutions. I have an enormous fondness for them because a Professor Meiklejohn, who was one of my mentors, once noted, actually it was over a glass of bourbon, that while universities and graduate institutions have only about 10% of the population, it's 100% of the future. And this is why institutions, universities, are really very important institutions to cultivate and to continue to support. With that, I'm going to take you on a story that is continuing, but it's in its final phases. And this is a story about a microorganism and the devastation that this particular organism caused on a particular population, and how using some more innovative developmental tools, a really substantive approach to controlling this particular problem has occurred over the last couple of years. To begin with, Martin Maiden is a professor of biology in Oxford, and Martin's been a very strong supporter of the project. And in fact, earlier this year, I gave a couple of seminars there under Martin's tutelage. And he put together a cartoon describing what happens to a particular organism. And this is the cartoon of the organism that is called Neisseria meningitidis. And this is an organism where humans are the only host. Doesn't live anywhere else except in humans and in the posterior nasopharynx of humans. And it's an organism that is spread by contact. And about 10% of the population is usually colonized with this organism. So in this group, there may be 100, 150 individuals here. There's certainly 10 or 15 individuals that are currently carrying fully virulent Neisseria meningitidis in the back of their throats. This organism circulates, as we say, pretty quietly in the population, and for reasons that are not terribly clear, becomes invasive and goes across tissue barriers, get into the bloodstream, get to the central nervous system, and then cause really a devastating disease. Now, the disease burden of this particular organism is very unusual. The organism causes disease globally, but 90% of the disease burden globally is in this belt that is called the meningitis belt in Sub-Saharan Africa. And in this particular belt, there are several strains of Neisseria meningitidis, but there's one strain that has been active in Sub-Saharan Africa for over 100 years and causes about 80% of the disease. And this is group A, Neisseria meningitidis. Every year there are outbreaks that occur, and I'm going to show you one of them. And what is particularly scary about this organism is about every 10 to 15 years, these epidemics coalesce into massive epidemics that sweep across Sub-Saharan Africa. Now, to put the area that we're working in some context, I just want to highlight just 4 or 5 things. First off, these are the poorest countries in the world. They have essentially no natural resources or very few natural resources. It is a very tough climate. People have subsistence incomes. There's no disposable income, and the per capita income in this area for many of the countries works out to be what I call dollar a day country. So it's between $300 and $400 per year. You know, That's not much money. And so consequently, the resources that are available are not excessive, I'll put it that way. I chose to show you what a typical epidemic that occurs every 12 years or so looks like in a district called Bousse district in Burkina Faso, which used to be known as Upper Volta. And this is a district with a population of about 130,000. I understand Clark County is not that far from 130,000. And I chose this specifically because the populations match up. This is the epidemic curve for an epidemic of meningococcal meningitis, group A disease, that occurred, and you could see that the disease occurred largely over about a month. And there were over 1,000 cases of acute meningitis that occurred over the course of a month, and giving you an incidence rate of 740 per 100,000 population. The average incidence rate for meningococcal disease in the United States is 0.3 per 100,000 population. So we're talking about something that is 1,000 or 10,000 fold higher than what happens in the United States. But I just want you to imagine what it would be like here in Athens if in 3 or 4 weeks you had 1,000 cases of acute meningitis. And for those of you who have either had meningitis or have had brothers or sisters with meningitis, you know how scary it is. This is not like a cold. This is a pretty serious disease. And in Africa, and this again is from the Bousse district this was 2006. There are no hospitals, and so people get treated where they fall. And they tend to sort of cluster in areas where there's a nurse. There are no physicians in these areas, and usually nurses manage all of this. And this is what a typical sort of epidemic would look like. This is a youngster who's just had a grand mal seizure, who's being cooled down by his mother with the water that's in this particular jug. This is a young boy who was, as best I could tell, somewhere between 11 and 12 years of age, who survived his bout of meningitis, but his problem is he's now stone deaf. And what's important to remember is that about a quarter of the individuals who are identified as survivors, that is, who have survived the bout of meningitis, are severely maimed as a result of that episode. Most are completely deaf, and they certainly don't have the number of IQ points after the illness that they had when the illness began. So what happens is you have this catastrophic event. All the stores and the markets close, people stay in the houses, they don't want to interact with anybody, virtually total collapse of commerce and interchange occurs. And so it's a major socioeconomic blow as well as being a major medical one as well. And when you put the numbers together, this is what this looks like. This is the epidemic curve for cases of reported meningitis. Not total, but those cases that ended up getting reported. And this is on the ordinate from 0 to 200,000 cases. And on the abscissa from 1950 out to 2010. And you see these repeated peaks that occur about every 10 years, and you have a major peak that occurs in 96, 97, where it was estimated that epidemic season there were about 250,000 cases of acute meningitis that occurred over a 6 to 8 week span in the belt. You add up all of these numbers and you get to over 1 million cases very quickly, just to give you an idea of the scope and the magnitude of this particular problem. Well, that's the background. And now what I want to do is to try to put in perspective what the situation was like in 2001 when the project began. In the United States, or I won't say in the United States, globally, most vaccines are made by a group of vaccine manufacturers that are called pharma. Pharma is the term that's given to this group of 5 multinational firms. These are very profitable companies. They use publicly funded basic research to develop their tools. This is publicly funded. They follow an expensive development process. They charge very high prices for new vaccines, and obviously they concentrate on low volume, rich world markets. Many of you received a dose of either Menactra or Menveo, which is the conjugate meningococcal vaccine before you attended college. That's one of the standard recommendations. And if you recall, that dose set your parents or yourself back somewhere between $100 and $150 per dose. Now, there's another group of vaccine manufacturers that actually ended up playing an important part of this story, and these are called emerging suppliers. This is a diverse group of companies that are located globally. They're not in Europe or the United States. Several of the companies are in India. It's an important home for them. Traditionally, they've sold less complex, simpler vaccines, and they've concentrated on what are called high-volume, low-margin markets. And these are the markets that basically pharma left. Pharma no longer makes DTP vaccine, which all of us received as infants and have moved on, and that gap has finally been filled by largely developing country vaccine manufacturers. However, these companies also recognize, what it's like to do business, and they're interested in developing more complex vaccines. Now, this situation then results in some problems for vaccines that are aimed at developing country problems. First off, development of new vaccines are controlled by pharma, and pharma wants to make products that have market potential. So there isn't very much interest in developing, because if you do your either MBA or any sort of accounting training, there is this thing called opportunity cost. If you're working on an inexpensive vaccine that's not bringing much revenue back, that occupies the time that you could be spending developing a far more profitable vaccine. So what happens is the development works against developing products for developing countries. And secondly, once products are developed that may have some applicability to developing country populations, usually takes an awful long time for the vaccine to percolate from developed countries like the United States, Europe, and Japan to developing countries. And I'll give you 2 examples: hepatitis B vaccine and Hib vaccine, that is the vaccine against Haemophilus influenza. These are 2 vaccines with a disease that has a global disease burden that took on average 18 years from the time it was introduced in the United States to the time it was used at public health scale in developing countries. So there's a long time that gets spent getting these vaccines developed. And I want to sort of point out one more thing in terms of vaccines and in terms of meningococcal vaccines, is that there are 2 families of meningococcal vaccines. One is what I would call the old family and the new family. The old family are polysaccharide vaccines, which means that you can grow these organisms in an incubator, and as the organisms grow, it sheds a carbohydrate capsule that's a sugar capsule that's on the organism itself. That sugar capsule is antigenic and is protective. But if you use that as an antigen, the antibody response, it tends to be short-lived. It's effective in adults, but is not effective in infants, and doesn't generate what's called herd immunity, a concept that I'll come back to later. But if you take the carbohydrate itself and you link it to a carrier protein, you then fundamentally change the immune response into what's called a T-cell dependent immune response that is much more powerful. It has memory. It actually has much higher antibody titers. Some of that information we'll show you a little bit later. So what we have is really two families of vaccines, and this is the family of vaccines that was relegated for meeting the African problem. In 2001, only these polysaccharide vaccines were available, and they were largely used in reactive campaigns. Now, I have to define reactive campaign. When an epidemic begins and really gets going, the reactive campaigns are based on what's called a ring immunization strategy, which means that the epidemic goes across a particular threshold. It is then reported to national officials. The officials then do bacteriologic studies, identify the strain that is causing the meningitis. Let's assume that it's strain group A, which causes most of the disease. They then contact the World Health Organization. The World Health Organization contacts the vaccine manufacturer. The vaccine manufacturer then tries to figure out where the money's going to come from to pay this particular vaccine. That vaccine then gets shipped to WHO, then gets shipped to the country, then goes from the country into the district where the epidemic is occurring. Do you get a picture from this? The picture is, remember I showed you the curve. You only have 6 weeks to work if you're going to do anything with a particular epidemic curve. So what happens is that most of these quote-unquote reactive campaigns, not most, almost all of these reactive campaigns occur after the epidemics. So that what happens is they're expensive because it's expensive to actually mount these particular campaigns. But from a public health standpoint, it's totally useless because it occurs after the epidemic. So you have now people are starting to feel a little bit better. They feel good because the government's showing up to give some injections. But the public health impact from all of that is virtually nil. So not a very good situation. Now, this was the situation that led to the creation of the meningitis vaccine project. And this was really quite straightforward. After the terrible 1996 epidemic that I talked about, African ministries of health as a group presented themselves to the World Health Organization and said, we really have to do something about this. We cannot continue with these reactive campaigns that don't accomplish anything. And so under WHO leadership, there were a series of international meetings, and one recommendation emanated from the 3 meetings, the large meetings that took place. And that recommendation was, we need to develop a conjugate vaccine, a better vaccine than the polysaccharide vaccine that we're currently using. That was the recommendation. And a collaboration came about with the World Health Organization and an NGO in Seattle called PATH. And a grant was submitted to the Gates Foundation, and in June 2001, MVP was created with a grant from the Gates Foundation. It was a very unusual grant. It was a grant for $70 million. It was a 10-year grant of $70 million where the money was given up front, so any interest that was earned went back into the project. And it was a collaboration with 2 agencies, that is PATH and the World Health Organization, with the goal of developing an affordable meningococcal conjugate vaccine for Africa. I was asked to join the project essentially in June when the grant was given, and knew about this and was very interested in the problem because I've worked off and on with this particular problem over a long period of time. And I realized when I went over the minutes for these 3 large meetings. The meetings took place in Geneva, Seattle, Washington, that is not Nairobi, Ouagadougou, or Dakar. And there was maybe 5 or 6 Africans that were there. They almost didn't say anything. And I couldn't find what the Africans wanted out of this. So very soon in July or the beginning of August, it became very clear to me after I'd gone to Geneva, I just looked over and read through the minutes and decided that we didn't know anything because this was an African problem. And so the first 9 months, and these turned out to be pivotal issues that came out from these meetings, we spent in Africa going country to country, meeting with epidemiologists, ministries of health, ministries of finance, EPI managers, physicians, etc. And what became clear were there were 4 issues that fell out of this work in 9 months. That is, epidemics was still occurring, that these reactive campaigns were very difficult and were not getting anything done, that there was very poor surveillance, that a lot of this stuff was guesswork and that needed to be strengthened, and that there were important conditionalities that applied to any vaccine that would be developed. And this is where the conversations got very interesting, because they then said that cost was probably the most important deterrent to the introduction of any new vaccine in Africa. And then when we explored this a little bit more, we said, well, all right, what makes it affordable? And the Africans hadn't grappled with that, but we went from ministry, and particularly with ministries of finance, et cetera, and it turned out that they said for a vaccine to be assuredly continuous within their budgetary planning, the vaccine would have to cost less than 50 cents a dose. This was very important at the time. I didn't realize it, but at that time, that established the ceiling price for the vaccine. Throughout 2002, we met with pharma, with the large vaccine manufacturers, and could not arrive at any agreement with them, particularly with a transfer price of less than 50 cents a dose. They said this was impossible, this never could be done, it's a lousy vaccine, you're crazy. I won't go over all the stuff that happened during those discussions. And what it required as we worked our way through this is that there would be a new strategy that would have to be developed. And that new strategy required MVP to reach out to these emerging suppliers. And it took us a couple of years, but, and I'm getting a little bit ahead of the story, but we decided after we evaluated some of these emerging suppliers that the meningitis vaccine project would become a virtual vaccine company. That instead of simply transferring the money to pharma, we would become a vaccine company and actually become responsible for the development of this vaccine. And this was the business model that we put together. And in Cartesian terms, you just take a big problem and René Descartes would say, take it apart and then look at the individual parts, because that will give you ways of solving these particular problems. And in a way, this was done quite formally. And we elected a triangular development process that isolated the development of vaccine-grade polysaccharides, and that was developed by a company in Amsterdam called Cinco Biopartners. We needed a supply of a carrier protein, and we chose tetanus toxoid. And Serum Institute of India knew how to make tetanus toxoid. And secondly, they were willing to accept the technology to make a conjugate vaccine and were willing to work under a price cap of less than 50 cents a dose. And the third arm is that you need a chemical way of being able to link the sugar and the protein. Now, all of, or many of these methods are protected by patents. Those of you who get into this, intellectual property is extremely important and highly protected. So we contacted pharmaceutical companies and asked whether we could use, and they, I think, quite appropriately said no. Get your own. And here we were very lucky, and it is an example, I think, of the United States government at its best. We were able to identify a conjugation method and help evolve the intellectual property and the patent for this at the US FDA, at the CBER laboratories at the US FDA. That was then transferred, or through the NIH has a group of the Office of Technology Transfer. And in 6 months' time, we were able to identify and transfer, with all the I's dotted and T's crossed, a new conjugation method from the FDA to the Serum Institute of India. As I say, it was an example of the US government at its best. And this is what happened. This is the vaccine that ultimately was developed. It was called MenAfriVac for obvious reasons, Men-A, free for Africa, and vac for obviously vaccine. And the vaccine then went into field trials. And here is just an example of the scope of the field trials that had to have been done. The two sponsors for the field trials were us, the Meningitis Vaccine Project, and we were officed in Ferney-Voltaire, right next to WHO in Geneva, and Serum Institute of India as the two sponsors. We then had CROs, and we wanted to make sure that we left something behind. And so rather than using large Western CROs, we chose an African CRO and an Indian CRO that continue to work with us. These were the test sites throughout West Africa and India. These were the sites and the laboratories that were chosen to actually analyze all the serologic work were scattered all over the United States, Norway, the Health Protection Agency in the UK. It truly was a global effort in terms of getting all of this done. And I'll just show you a little bit of the data. We have reams and reams of data that are now beginning to be published. This comes from a large publication that looked at the immunogenicity of this vaccine in 12 to 23-month-olds, 2 to 10-year-olds, 11 to 17-year-olds, and 18 to 29 years. And this is important because these were the target populations that we had for use of this vaccine in large mass vaccination campaigns. And it compared two vaccines, the conjugate vaccine that was called PSATT, and that's the polysaccharide that is complex to tetanus toxoid. And this is compared to simply the sugar vaccine, which is the licensed vaccine. And if you look at pre and post with the polysaccharide vaccine and pre and post with the conjugate vaccine, that is these black bars, outperforms the polysaccharide vaccine in every age group, and gender didn't make a difference, nor did site. These were very, very clear data. And these results are in log frames, so that the SBA titers, this is 1 to 10, 1 to 100, 1 to 1,000. So this difference, these are huge differences. These are easily 10-fold differences. So we were very relieved that the vaccine was highly immunogenic and was safe. And when all of these data were analyzed in terms of the potency and safety of the vaccine, if administered to 1 to 29-year-olds, there were 3 conclusions that immediately became apparent. The vaccine was well tolerated and it was safe. Secondly, it was highly immunogenic and it was superior to the licensed polysaccharide vaccine. It induced immune memory, and it also when we followed the titers in individuals out to 2 and 3 years after they have received a single dose, the antibody titers were sustained. Lastly, it also, because the carrier protein in this vaccine, tetanus toxoid, was also immunogenic, the vaccine behaved as a bivalent vaccine, and you also got for free a dose of tetanus toxoid. After a lot of work, the vaccine was, or the dossier was prepared, and I should have brought a picture of what the dossier looks like. The dossier was 13,111 pages in what is called the Common Technical Document. This is a bear. I know because I read most of the pages of this document. They're very strict rules, as they should be. But when you finished all of this work, it took 14 months to actually generate the Common Technical Document. This then led to a license from the Drugs Controller General of India in December of 2009. Then the dossier was submitted to the World Health Organization for a global license, and WHO granted pre-qualification of this vaccine in June of 2010. June of 2010, almost all of the epidemics in Sub-Saharan Africa occur from about the second week in January through April. So with the vaccine licensed in June 2010, we started thinking that if we worked hard enough, we would be able to mount an immunization campaign in December of 2010, just before the epidemic season. And so after a tremendous amount of work, this is what happened. This is on December 6th of 2010. The vaccine was introduced. This is Blaise Compaoré. This is the president of Burkina Faso, appropriately coming down the red carpet. And he was instructing the country of Burkina Faso that he wanted the vaccine to be, he wanted the entire 1 to 29-year population vaccinated. These were the vaccinators, and it's hard for me to describe how exciting this was. This was done in one of the large centers in the middle of Ouagadougou in a horseshoe arrangement with maybe 5,000 school children within that particular horseshoe. And then there was the Director General from WHO flew down, the representative from UNICEF, Gavi, the World Bank, Gates Foundation sent a large retinue of individuals, and it was a phenomenal day. And I always remember the president's speech, and he gave it in 2 local languages and in French. And I'm fluent in French, so I was able to understand exactly what the president said. And it was fascinating in that he challenged all of the school children and said that this was a once-in-a-lifetime opportunity for Burkina Faso to rid itself of this particular disease. And he wanted every single child that left that auditorium, not auditorium, but that open area, to get vaccinated before they went home. They had to go home with a vaccination card. And that there were vaccinators in every single school in Ouagadougou that particular day. But you could have heard as he's speaking to the school children a pin drop. It was really astonishing. And then what happened is the vaccination campaign unrolled. And this is a typical Burkinabe being immunized. This is a typical clinic with, this is the cold box with this, there's the vaccine, and individuals are being vaccinated. The target population, 1 to 29-year-olds, make up 70% of the population in Burkina Faso. So you can see how young the populations are, 70%, 1 to 29-year-olds. And they decided that they would do the whole thing in 10 days. The target population in Burkina Faso was 10.7 million individuals. The campaign lasted 10 days, and they immunized 10.8 million individuals. This was an extraordinarily successful vaccination campaign with very high acceptance. Very high acceptance because individuals understood that this was a new vaccine, and when they would talk amongst themselves, they referred to the vaccine as le bon vaccin, that is, the good vaccine. And that was the term that was used in the marketplace, that this was le bon vaccin. And so the turnouts were enormous in terms of being able to get coverage that easily was above 95% for the entire country. So it could not have been done better than what transpired in Burkina Faso. And this is what happened. This is the epidemic curves in Burkina Faso. Remember I told you about the epidemic in 2006-07 in Bousse. These are the large epidemic in Burkina Faso from 2006, 7, and 8. This was a total of, I think it was somewhere along 48 or 50,000 cases that occurred. And then here's the introduction of the MenA vaccination campaign in December of 2010, and this is what happened in 2011. At the end of the 2011 season, we plotted the cases of meningitis by bacterial isolate. And the bacterial isolates are, again, NMA, which is Neisseria meningitidis group A. That's what the vaccine was targeting. And so you can see plenty of disease in Chad and in the Cameroon. Nigeria, we didn't have any data. This is Mali, this is Burkina Faso, this triangular cap, and this is Mali, this is Togo, and the distribution of pathogens was such that if you look at this, you don't see any red in Burkina Faso, Mali, and Niger. These were the three countries that were immunized. We did all of Burkina Faso, part of Mali, part of Niger. But we obviously didn't have enough vaccine to do Chad and Cameroon, nor enough resources for that matter. And so we were very, very pleased to see no red in these particular countries. And then when we analyzed the data much more carefully, this is what came out. There were 3,875 cases of meningitis that were reported. In an active year, that's usually about 30,000. And so this pretty much represents what occurs naturally, that you're unlikely to change that, at least with a MenA vaccine, maybe with other vaccines. And the nurses were able to get 3,125 spinal taps. And we looked at the spinal taps. There were some that were contaminated, some that were negative. 1,000 were positive, and when we looked at the rate, it was about a third. In the United States, for a suspect case of meningitis, that's exactly what we find. About a third of cases are identified that come in that are worked up with a lumbar puncture. So we felt that this was probably very representative. And then when we looked at the distribution of pathogens and used only the positive samples as the denominator, we found almost no group A disease, an unusual strain called Neisseria meningitidis group X, an enormous number of pneumococcal meningitis that had always been there but had never been recognized because they had been covered up by all the group A disease that occurred every year. And when we put this together, this is what these data look like. Again, these are the years from 2005 to 2011. This is this big epidemic that I told you about with 20,000 cases, 2006, 26,000 or 27,000 cases, 2007, and 10,000 cases, 2008. And if you look at the fraction of MenA that were isolated, it's almost 90% of those isolates are group A disease. So that's a splendid example of how serious the MenA outbreaks are. Then we introduced MenAfriVac, And look what happened to the fractional distribution of Men-A. It went from 25% in 2010 to less than 1% the following year. And when we looked at those 4 isolates, what was interesting about it was that 3 of the 4 isolates were individuals from Togo that developed meningitis, but that the healthcare services were so bad in Togo that they were brought into Burkina Faso for treatment, so therefore had never been immunized. And when we subtracted out, we found only 1 indigenous case of Group A disease. This was in a 17-year-old girl who lived in the desert area between Mali and Burkina Faso, and we're not sure which side of the frontier she was on, but she was never immunized. And so all of 2011 resulted in 1 case of Group A disease. We are now about halfway through the next epidemic season in 2012. There has been 0 cases of Men-A so far this year. So the impact has really been quite pronounced. Now, at the same time, we were interested in what's called herd immunity. And herd immunity occurs this way. If, as I said, you're colonized with Group A Neisseria meningitidis, and if you get immunized and you can either decrease the length of time that you carry, in other words, if the vaccine is therapeutic or curative from your carrier state, then what happens is the organism doesn't have anywhere to go. And in epidemiologic terms, they talk about R0 falling to less than 1. And when that happens, the organism literally disappears from the population. This is what we want, and this is what has happened in the UK with a monovalent Men-C disease. So we felt it was very important to study this particular issue of what happened to carriage before and after immunization. So we worked with the CDC and the Norwegian Institute of Public Health and designed a very large carrier study. This is what the carrier study is like. I mean, it's just, if you've got any kids, you know what this is like. I mean, it's the same thing as taking like strep throat, only this time you're looking for Neisseria meningitidis. And we studied 2 rural areas and 1 urban area, this is what happened. This is the percent of isolates from these throat cultures that were positive for Group A disease before and after immunization. Before immunization, we had these 3 districts. One district called Kaya had a pretty high carrier rate of Men-A at about 1%. The second was Dande. Dande was somewhere around 0.3, 0.4%. And Bogodogo is an urban area in Ouagadougou, and the carriage was never very high in that area for reasons I don't particularly understand. But when we put together the data from 20,325 throat cultures taken over 5 sampling intervals, we found an overall carrier rate of about 0.4%, which is very consistent with what the Brits found with the Men-C carriage data in the UK. Look what happens post-immunization, though. This number is now 21,000 something. I've forgotten what the last 3 numbers are, but it's about 21,000 samples have been taken now in 4 large serial sets of cultures in the same population. Not 1 Group A Neisseria meningitidis has been isolated, which meant that the organism has disappeared from the population because of the genesis of herd immunity. And the results from the impact observations was that there was a dramatic reduction in reported meningitis. There was disappearance of Group A, and now the absence of Group A in carriage studies, and all consistent with vaccine-derived herd immunity. And we asked ourselves, you know, you work 9 or 10 years on something, and you don't know until you know. We actually were started asking ourselves, why did this work so well? You know, it's almost like all your prayers are answered. You know, you've been working on something, and then, you know, usually nature isn't this generous. You know, and so we went through all of the data. We had all of the cultures reconfirmed at the National Institutes of Public Health in Oslo, and really tore through all of these data saying, did we get something wrong? And then in sessions with epidemiologists, we realized that we probably didn't get it wrong. That this was a very potent vaccine. We had very high carriage rates, over 95%. And the background rates of carriage were pretty low, and they were probably pretty low because of the epidemic that occurred in 2006, 2007, and 2008. So what we did in 2010, we think, is we gave a dose of vaccine to what's called a primed population, a population that's already had an intense exposure 3 years before with this particular agent. So they were primed, and this particular dose caused antibody titers that were extraordinarily high. And the antibody titers were so high that the vaccine functioned as a therapeutic vaccine. And this is what we think happened, and I'll come back to Martin Maiden's cartoon, is that each one of these potential areas for the organism to go became blocked as you increase this high immunity in all of these particular populations. So what happened? Post-2010, as I said, we had done about half of Mali, Burkina Faso, and Niger in the fall of 2011, that is just before this epidemic season, was our second season. We immunized the rest of Mali, the rest of Niger, 5 states in northern Nigeria, and northern Cameroon, and a corner of western Chad. If you're trying to get herd immunity, it's important to get the geography together so that the protected populations are contiguous. And this way, the organism doesn't have a hole to sort of drop into an unprotected population. And so that's what's transpired this year. The results have been excellent again through this year. There's essentially now no MENA. I don't know about Nigeria. We don't have data from Nigeria, but we certainly have now data from western Chad. The organism has disappeared, and the organism has disappeared from Cameroon. And the results in Mali, Burkina Faso, and Niger are the same. That is, no disease. Let me finish with just one observation. And remember I said that the length of time that it takes for vaccines that are developed to get to developed countries. We talked about hepatitis B and Hib vaccines the year that they were licensed in the United States. And when you work it out, you know, it took 18 years for scale up into countries, into developing countries, and 19 years for hepatitis B. One of the things that we're most proud of, if you look at the development of the conjugate vaccine, MenAfriVac, that occurred in 2010, and that was used at public health scale in 2010. As of now, that is this particular date right now, 55 million Africans have been protected. Our target is, we'll work out to somewhere between 250 and 275 million to actually finish all the primary vaccination programs. And that will finish probably sometime in 2016. But within the first 2 years, well, actually within the first 18 months of having a vaccine licensed, 55 million Africans have received that particular protection. And none of this is possible, obviously, without partners. I haven't spent a lot of time talking about partners, but these are the sort of gang that really all work together in terms of getting this vaccine developed and introduced at public health scale, including, you know, the CDC, HPA, Gavi Alliance that actually has given now $130 million in introduction costs to see that this gets done. Obviously, WHO, the FDA, Serum Institute of India, PATH, et cetera. None of this is possible without the collaboration of just a lot of people. And I'll finish with this individual. If any of you were able to visit our office in Burkina Faso, you walk in and this is the guy that you see. Right at the very beginning of the project, we wanted to make sure that the focal point of all the work that was done was the African. It wasn't Serum Institute of India, it wasn't WHO, it wasn't the Gates Foundation. Decisions were predicated on what was best for this kid. And when that was done, and that's one of the most important lessons of all of this, is when everybody agreed to that, then all this crap, all of these problems, all of this other stuff just disappear, because everybody gets focused on the real issue. The real issue is making sure that this individual isn't maimed, killed, or made deaf. Thank you. Right. Yeah, three great questions. The first is, well, okay, now that you've done something for A, what's in the pipeline? And that's exactly what the next step is. The next step is that we move to a more complex vaccine. Instead of a monovalent conjugate vaccine, which is relatively easy, although easy in parentheses, then we need to develop a polyvalent meningococcal vaccine. And so that work has begun, and that work is in progress. Secondly, what about booster vaccines? This stuff all looks pretty good, but what's it going to look like 5 years from now, 10 years from now? Well, we do know on the basis of the British studies that the MenC immunogenicity and herd immunity has lasted 13 years after a single dose of a MenC conjugate, which I don't think is as potent as this particular vaccine. But the correct answer to your question is we don't know. And so that what we need to do is to maintain surveillance and maintain serologic surveillance in these populations. If antibody titers are to wane over a period of time, then we may want to consider our booster doses necessary. So what I would say the correct answer is time will tell with that. The third question said, what happens in HIV populations? You know, I don't know. We specifically excluded HIV populations from any of the studies that were there. And when we finished all our licensure studies, frankly, we did not have enough money to go back and now specifically look at HIV populations. So that what we have is a protocol that's up to funders now, that we're looking for funding to do 1 or 2 trials. One, we think can quite easily be done in Dakar. Okay? Questions?
We didn't exclude HIV.
No, we didn't exclude HIV, and that was in consultation with the World Health Organization, et cetera. There's very little data on meninge being as a bad actor in HIV-positive individuals. It's not like pneumococci. Pneumococci is very good data to show that you really are at increased risk. With meninge, that point has not yet been established. However, you know, it stands to reason that if you've got problems with pneumo, you're likely to be not as robust in terms of your immunogenicity against meninge. So I think it's it's essential to get individuals who are HIV positive, frankly, vaccinated.
Do you think their antibody titers may fall off?
In point of fact, when I actually look at the data, the immune response in the very young ones that have never seen MenA are spectacular. Well, you saw those there. And so what I've posited is just a theory. It might work out that this is just a very good vaccine, and you know, as somebody from WHO said, well, you know, I wouldn't hurt myself over the fact that it was a good vaccine. And so, but I think that question and other questions are going to be gradually resolved as we continue to sort of churn and to get data. And this is where the CDC is just an invaluable ally. And again, I say this, you know, really with heart and hand that, you know, when the U.S. government wants to do something, and unfortunately, it's been distracted for the last several years, but when it wants to get its stuff together, this is the kind of work that can get done. You know, it's really amazing. And I say CDC, it's obviously the Norwegians, it's the French, it's everybody that gets together.
Other countries that you've since moved into?
Yes and no. It's not been the 95 to 100%, but it's been above 90%, which is astonishing. And when we've had, I believe in focus groups. You know, when things like that happen, I love to sit down with people and just schmooze with them. And that's how you understand a little bit what their perceptions. And the perception that came through in meeting after meeting with mothers, fathers know nothing. But mothers really know what's going on. And their sense was they were so tired of this. And they would phrase it in things like this that would really grab you in the heart. You know, the epidemics are such that when the first rains come, it stops immediately. It absolutely stops immediately. As soon as the relative humidity after the first major rainstorm comes, disease, even if you're in the midst of an epidemic, everything stops. And the mothers call that the rains of hope. I mean, that's what they'd say in their own language. That was the rains of hope. Now you sit down and talk with them. Well, what's the rains of hope? Well, she said, you know, every year we have to worry about this. Is this going to be the year? Is this, you know, is it going to come? Because when it comes to these compounds, it may take one compound and 4 kids in that compound get meningitis. There's a compound 50 yards away, nothing happens in that one. And then about 200 yards away, there's another 3 or 4 that come down. It's extraordinarily focal and unpredictable, so that mothers, you know, have this dread. It's almost Kierkegaardian, you know, this sort of fear that occurs. And they verbalize that. And so, and their response would be, get the vaccine. We'll take that vaccine. We'll get our kids to take our vaccine. We'll get our husbands, we'll get our children to get that vaccine. And they were right. So I think it was just a general fatigue of that disease burden that really prompted this, and it wasn't any sort of genius or anything. I think that people are just sick and tired of this. Yeah.
That needs to be identified.
Yeah. No, that's a very good question. And in point of fact, when we've put together the things that have worked with this project and the things that didn't work, one of the things that didn't work is I could never get universities interested in studying exactly the same question that you're bringing up, which is, what is the factor? Why does this go crazy? You know, it does nothing for 7 or 8 years, and then all of a sudden you have these tremendous attack rates. What happened? I mean, you know, is it the dust? Is it a rhinovirus or an echovirus, or what is it? And to this day, we don't know. And it may be an instance where this vaccine, no, not this vaccine, but this disease, if we're going to be lucky, will largely disappear, and we never will have answered the basic question in terms of why. You know, that's a good, you know, pneumo is, that's a big deal also. That's another story.
So let's continue this conversation. Please join us next door. We have food and drink available. Thank you.
I'm going to get that out there. Thank you.